Hello everyone, welcome to Biomixup. In this video, we are going to discuss about the basic of transcription. Before starting about the transcription, we should definitely know about the basics. What is transcription, enzymes which are involved in transcription, and where this transcription is taking place. It is taking place in the prokaryotes or in the eukaryotes. Is it same or it different? Let us first see what is transcription. We can define transcription as a process by which RNA copy of a gene is made from a DNA. That means if we are having a DNA, from that a RNA is produced from which we can synthesize the protein. Let us see with this diagram. See, here we are having a double-stranded DNA. This whole total is known as central dogma. First of all, what happens? We are having a double-stranded DNA. If from this double-stranded DNA, another double-stranded DNA will be synthesized in presence of the enzyme DNA polymerase, it is known as DNA replication. If from this single strand of a DNA, which will work as a template, a RNA is produced, this process is known as transcription. The process of formation of a RNA from a DNA strand is known as transcription. Okay, now from this RNA, if protein is produced, it is known as translation. How this protein will be produced? The codons which are present in the RNA, three codons will be present, three triplets will be formed for one codon. This codon will be responsible for some of the amino acid. In this way, various types of amino acid will be produced, and these are the single amino acid. When various type of amino acid will be binding together, they are known as protein. So, this whole total is known as central dogma. First, we have to know about the transcription. Before discussing about the whole process, we have cleared that what is transcription. The formation of a RNA from a DNA is known as transcription. So, how we will differentiate that whether it is a DNA or it is a RNA? The major difference is between the base pairing. What happens? There are few bases. They are the purine and pyrimidine. They are A, T, G and C. This A means adenine, T means thymine, G means guanine and C means cytosine. This adenine will always base pair with thymine, and the base pairing will take place in presence of two hydrogen bonds. Next comes the guanine. This guanine will always base pair with cytosine and in presence of three hydrogen bonds. This is in case of a DNA. So, what is the difference between the DNA and RNA? The major difference is between the base pairing. There is another nitrogenous base which is known as uracil. This uracil is always present in a RNA. This adenine will base pair with uracil in presence of two hydrogen bonds. That means thymine is being replaced by uracil in case of RNA. This is the main difference to differentiate between the DNA strand and the RNA strand. We have discussed about this line. What is transcription? It is a definition. If you want to note it, please make this video pause and note it down. Now, let us see some other examples. See here, we are having a DNA. If from this DNA is synthesized, it is known as replication. If from this DNA, RNA is synthesized, it is known as transcription. And if from this RNA, a protein is produced, it is known as translation. We have known the basics of this transcription. What is transcription? We have discussed. We have to now see where this transcription is taking place. In case of a prokaryotes and in case of a eukaryotes, the place is different. But why the place is different of the synthesis of a mRNA or a RNA? Because in case of a bacteria, that means in case of a prokaryote, there is absence of a nucleus. That is the reason everything will take place in the cytoplasm. See here, the DNA is being synthesized into DNA in the cytoplasm, and the transcription is also taking place in the cytoplasm, that is the formation of RNA, and the synthesis of protein, that is translation, is also taking place in the cytoplasm. But what happens in case of a eukaryote? The process will take place in the nucleus. The DNA synthesis and the replication and the transcription, that is the formation of a mRNA, will take place inside the nucleus. Replication and the transcription will take place in the nucleus, and after the modification it will be transported into the cytoplasm. Then in the cytoplasm the protein will be synthesized. This is all about the difference between the prokaryote or the eukaryote. Here it is another diagram. See here also it is an example of prokaryote. Everything is taking place in the cytoplasm, and in the eukaryote DNA synthesis, mRNA formation, and the processing is taking place in the nucleus, and the formation of protein or translation is taking place in the cytoplasm. Okay. So the next thing which we have to know is the direction. In which direction this RNA is being synthesized? It is being synthesized in the 5' to 3' direction, because it needs a 3' OH group. Okay. So the DNA is having two strands. Okay, here there are two strands, but one will act as a template for the synthesis of RNA. Suppose this strand will act as a template. This will act as a template. So by using this DNA strand, RNA will be produced. It will copy, or the complementary base pair will be produced by using this template, and this template is known as nonsense strand or non-coding strand. Non-coding strand, and the strand opposite to it is known as sense strand. It is known as sense strand or coding strand. Okay, and the major difference is between the U and T. In case of a DNA, T will base pair with A, and in case of RNA, T will be replaced with U. That means A will base pair with U. Okay. So there are some other terms like monosystronic or polysystronic. If we discuss about the monosystronic, that means only one type of protein will be produced from this mRNA or RNA. The structural gene will code for only one type of protein, and it is common in eukaryotes. When we talk about the polysystronic, that means a single mRNA will produce three different types of genes or proteins. It is common in prokaryotes. In the process of transcription, what happens? mRNA is produced or RNA is produced. After maturation, it is known as mRNA or mature RNA. After that, it will produce several proteins, and it is common in case of prokaryotes. So we have to know from where the reaction will take place or from where the transcription will take place, and which enzyme is responsible to start this transcription. For the transcription, there is an enzyme known as RNA polymerase, which will initiate and it will help in the process of transcription. And in which site the process will take place? Like in DNA, we are having a ORIC. In case of a transcription, we should have some of the site from where the transcription will take place. It is the promoter region where the transcription will start. Before the start codon, there is a promoter region where this RNA will come and bind. Next to the promoter is the transcription start site. That means there is a starting of the transcription, and after that there will be a start codon and a stop codon from where the mRNA will be synthesized. Okay. Next to the stop codon, after some reason, here there will be a UTR region, and here what happens, there is no synthesis of the mRNA. Let us clear with some other examples. Say here we are having a DNA strand. This is the DNA strand and it is a recognition site of the promoter. This is the recognition site where the RNA polymerase will come and bind. Here this is the RNA polymerase which came and bind in the promoter region. After the binding, what happens? It will open this DNA strand by melting the hydrogen bond. See here, you can see that this DNA is now open and this single strand will be formed. This RNA polymerase will help in the formation of mRNA from this single strand. In this manner, the mRNA will be synthesized continuously. Okay. Now the recognition site for promoter will be there. So it is the -35 element and the +10 element. Here there are some parts of the RNA polymerase. Here in the RNA polymerase that there are some parts which will specifically bind to the -10 and -35 element. Suppose this is the promoter region. How this RNA polymerase will recognize it? It will recognize because it is having the binding affinity with these regions. Okay, it is the TATAT box which is known as -10 element. -10 element is also known as Tata box. Okay, it is known as Tata box, and the -35 element is known as TTGAC box. TTGACG box. Wherever it will find this regions, this RNA polymerase will come and bind and help in the transcription. After this region, after this -35 element and the -10 element, there will be +1 site from where the transcription start site begins, and from this region the transcription will be produced or the formation of RNA will be produced. Okay, so here we are saying that it is written that it is a bacterial promoter. In case of a eukaryotes, we were having -35 and -10 element. Okay, but in case of some bacteria, what happens, there is a additional UP element or up element. There is no 35 segment. -35 segment is somewhat present in some of the bacteria and -10 is also present. This -10 is also known as Primno box, and in some case the -35 element is absent, only the -10 element is present and additional to is up element. Okay, but in some cases some lack -35 element but have extended -10 region. That means here see, we are having, in this case we are having -35 as well as -10 element. Okay, in this case we were having UP element, -35 element as well as -10 element. But in this case we don't have UP element or up element or -35 element. We have only -10 element but in the extended form. See here it is being extended. This -10 form is being extended. See here again we have a diagram, and these segments are known as a -35 segment and the -10 segments which are present in the promoter region. They are known as consequence sequence. Okay, that means it is being reserved. This is known as Tata box and this is known as TTGA box, and the difference here is minus or plus 17 base pair. Okay, here we are having +1 region. After this +1 region only the transcribed region is present, and from here to here till the +1 region from the upstream it is known as promoter region. So by this diagram you can be very clear about the coding strand and the template strand. See here we are having a DNA. From this DNA one will act as template strand, another will act as coding strand. Here this DNA is working as a template and it is producing the RNA. See, T is base pairing with A is base pairing with U as it is the RNA, C is base pairing with G, T, A. So this is the template strand. The other name for it is anti-sense strand. Okay, that means it is not sensing. The strand which is opposite to the template strand is known as coding strand or sense strand. Why it is named so? Because see the difference here. Here it is in the alternate way. But this strand which is known as sense strand and the RNA is same except this T and U. In place of A or T, U will be present and other things are already same. See, here we are having A, here also A, leave this T, G, G, A, T, leave this. In place of T, we will be having U in case of a RNA, C, C, again T, U is difference. That is the reason it is known as sense strand. That means it is same as the RNA except the T and U. Now, we have got the RNA and from this RNA there is synthesis of various types of amino acid. Here methionine is a amino acid, isoleucine is amino acid, serine is a amino acid. AUG stands for methionine which is a start codon, AUC for leucine, CGU for serine. In this way various types of amino acids will be produced and the amino acid will be linked by peptide bond. These are the peptide bonds by which two amino acids are being linked. They are known as protein. Okay, when small 2 to 3 it will be synthesized, it is known as polypeptide. When more amount of amino acid will produce and they will be linked, they are known as protein. So here we can see that there are primary RNA transcript and spliced RNA. What happens when the RNA is produced and it is not mature? Both exons and introns are present. What is the difference between the exon and the intron? Introns are the non-coding strand. Introns are non-coding. That means no amino acid will be produced from the introns. And what is exon? Exon means the coding strand. What happens when the RNA is produced? It is present in the nucleus and after the processing only it will be transported into the cytoplasm. How it will be processed? It will be processed by splicing, 5' capping and 3' polyadenylation. What is splicing? The removal of the introns, that is a non-coding strand from this total RNA, is known as splicing. The introns are being removed and the exons are being ligated. See here we have exon 1, exon 2, exon 3 and these are being ligated. And after this ligation, for preventing it from degradation by some RNAs and for the helping them to be transported into the cytoplasm, they need some modification. What are the modification? The modification is 5' capping and 3' polyadenylation. After this only they will be transported into the cytoplasm. So we have discussed about the basics of this transcription and in the next video we will discuss about the RNA polymerase, the structure of RNA polymerase and its description. And thank you for watching this video till the end, and if you like this video please do like, share and subscribe. Thank you. See you in the next video.